Hi everyone, welcome to my channel Pure Biology, and this is another video that is the role of the floral organ identity gene. Okay, so this part, this topic related substances, that is long, so I just divided into two parts. Okay, two parts. So here the role of the floral organ identity gene. These genes directly control the floral organ identity. Here, these genes generally control the floral organ identity. And the proteins encoded by these genes are transcription factors that interact with other protein cofactors to control the expression of downstream genes, and whose products are involved in the formation or function of the floral organs. So what is saying that? First of all, the proteins encoded by the genes are the transcription factors. So main important proteins are the transcription factors. These transcription factors first interact with the cofactors. These are another proteins. And in that way, by attaching with the cofactors and the transcription factor, both form a complex and that control the expression of some genes, downstream genes. So these downstream genes, whose products are involved in the formation of the floral organs or the function of the floral organs. So that means here the protein transcription factors and the cofactors both are the proteins, and that form the complex and that activates the expression of some downstream genes. By the activation of these genes, the formation of the floral organs is done. Clear? Now the flowers of the here one example has taken, that is the Arabidopsis. And this consists of the 4 concentric holes or organs. That what are that? These are that 1 to 4, the one corresponding to the sepal, and in that way the second one that is related to the petal, the third one that is related to the stamen, and the fourth one that is related to the carpels. So all flowers are that constructed along the similar lines and made up of concentric holes. In these 4 concentric holes that produce a flower or the parts of the flower, that is always same in every flowers. That 4 holes are needed for producing the 4 organs of the flower. Clear? And the 4 holes produce the same way, sepals, petals, stamen and the carpels respectively, and within this or among these the carpel is located in the center of the flower. You all know that it is located in the center of the flower, and the floral parts are each derived from a single hole of cells in floral meristem. So what is saying that? The 4 holes are there and each hole is related to particular one organ of the flower. That means that each hole's meristematic tissue, that means the floral meristematic tissue, that divides to form the particular organ of the flower. And the expression of homeotic genes in this result in the production of the transcription factors. So here important role is played by the homeotic genes, and these produce some proteins, obviously that transcription factors, that control the phenotype and organs. This is the organs, okay, and the organs derived from them. So important thing is that here the homeotic genes that play an important role for the production of the organs. Okay. Now this model is known as the ABC model. ABC means ABC, these are here that identified as the genes. Okay, A is one kind of gene, B is another kind of gene, and C is another kind of gene. So these are all the homeotic genes. Clear? And that determine the organ. So this is called the ABC model. Now the first hold, this is the one, okay, this is determined to be sepals by the product of A genes, okay, that is product of A genes. This is the AP1 and AP2. AP1 and AP2. Here I have written the short form only because AP1 and AP2 and another gene's full name that is already described in my previous video, so you just check out that one, okay. So AP1 and AP2 alone, these two genes are responsible for determining the sepal formation, clear? And which hold is involved in the sepal formation? The first hold, okay. The second hold, that is determined as the petals, and here the combined actions needed. Which genes are involved here? A genes and the B genes. B genes means AP3 and PI. Okay, AP3, PI. So A genes means AP1 and AP2, and B genes means AP3 and PI. So both A genes and B genes both activates to determine the second floral organ, that is the petal. And the third hold, that is determined as the stamens, and here the B and C genes are involved. So B and C genes means, B genes mean that is AP3 and PI, and C genes means AG. So B and C both genes activations are needed for this determination of the stamens. And which hold is related to the stamen formation? The third hold. And the hold 4, that is determined as the carpel, and here only C genes, that means AG genes, that is needed, or its activation is needed for the formation of the carpels. So here that is A gene for the sepal, and hold 2 A and B gene, AP1, AP2, AP3 and PI, this is the petal, clear? And B genes, hold 3 B genes, AP3 and PI, and C genes AG, so this is stamen, and next one C AG, this is carpel, okay. So here you can see the 4 holds are there. First one is the C gene, second one is the B and C genes, third one is the A and B gene, and fourth one is the only A gene is responsible. And you can see that how these organs are formed from for the expression of these genes. This is the carpel, stamen, petal and sepal. Okay, I just have to write these genes, so you just wait for a few minutes. Okay, I have written here the full name of the genes. AP means apitala, PI means pistillata, and AG means agamus. Okay, these 3 genes are involved for the floral organ formation. Its continuation process is there, but I have to explain that topic in the next video. So you just continue with my channel. Okay.